Okay, 11:01. I guess we can start in the East Coast. Um, Welcome everybody to our another exciting meeting of Myeloid Network. We are privileged to have Leila Akkari join us today with her presentation. Just a couple of words about her. She get her Ph.D. from CNRS in 2010, did the postdoc at Memorial Sloan Kettering, and then did some stint in Lausanne University, a relatively short one. And after that, in 2017, she is in Netherlands Cancer Institute when she grew up in rank and currently associate professor. What Leila is known for now, quite widely, her very interesting work on interaction between myeloid cells, primarily macrophages, and tumor in liver and brain cancer. She had very interesting, couple of very good and strong, interesting stories published a couple of years, last year and the year before, dissecting the mechanism regulation of the cells. And she utilized very complicated and sophisticated mouse models, which is really quite telling and important. And as a result, despite her relatively young age, she's already got recipient several awards and Dutch Cancer Society Young Award in ERC, Synergy Grant from European Union, and award in fundamental research from AMODA. So I believe it will be quite exciting time we spend together for the next hour. And as always, I'm pretty sure people who attend this Myeloid network regularly knows that please put your questions to the chat. I will do my best to ask all of them, or me or Jelani, or depending on how many questions you have. And we will work on this maybe even beyond the hour if time allows. Leila, thank you very much for joining us, and the floor is yours.
Thank you, Dimitri. Thank you to all the Myeloid Network for the past few years of really exciting seminars that I have followed as many as I could. Very interesting, different, diverse type of science that have been exposed, and I hope I will be up to the task today. Thank you for that very generous introduction. Indeed, my lab has been now at the NKI for about 8 years. We're also a member of the Oncode Institute. And today I just want to tell you about some of the work we've done in the past few years and give you hints on where we're going for the next ones, trying to unveil the determinants of myeloid cell heterogeneity in cancer. So we all know that the tumor microenvironment is an elaborate ecosystem. We've learned a lot in the past few years by many of the people present in this meeting and that really started this meeting, in how the stromal and immune local and systemic environment differently affect the progression of cancer and organizes within that environment in a very precise manner to fuel this malignancy. And so we've realized that despite an original good intention, the path to cancer, to macrophage, particularly phenotype in cancer, is well paved with good intention, ultimately supporting tumorigenesis. And so that's why we know that those immune and stromal cells are educated and hijacked to promote cancer progression by manifold, multifaceted complexities in the tumor microenvironment. Where we're still relatively poorly grasp is how this evolution of the tumor microenvironment co-occurs together with cancer cells changing in the course of malignancy, but also as the therapeutic intervention surely fails in most solid tumors. So for a long time, of course, those immune cells that abundantly populate this ecosystem were thought to be relatively simple targets because they were genetically stable. But we have realized as a whole field that it's a lot more complicated than that, and that this transcriptional plasticity, particularly in phenotypic plasticity, immune and stromal cells present, is extremely difficult to hijack, to therapeutically harness in solid tumors. And so up to now, the poster child success in immunomodulation remains T-cell-centric immunotherapies, and that is, of course, limited by the fact that in most solid cancer, there's not so much of those T cells. What there is plenty of is tumor-associated monocytes and macrophages. And again, work from this field has shown the potential of these cells as anti-tumor actors through depletion strategies and force reprogramming, mainly in preclinical models, showing very clear success of targeting macrophages. And yet we have not bridged the gap to apply those kind of approaches in patients and in clinical practices. And so I don't think that this is a secret for everyone, that we and others believe that this is rooted in the immense plasticity and versatility of these cells. And all this versatility is really hacked into the physiology of macrophages and monocytic cells in the different capacities that they have to maintain organ homeostasis and how rapidly they can adapt their phenotype when this homeostasis is disrupted, participating to it, while also being key central actor in restoring homeostasis. And this is through interactions within their environment, very precise organ-specific functions, but also all these different phenotypes that they can adopt that really ranges from phagocytosis to angiogenesis support, immunosuppression, immune activation, and metabolic regulation, which is becoming more and more prominent now in the context of cancer. So if we think about these different complex features that have influences macrophages, but in general immune cell functions, we like to think about it as different layers of complexity, one being intrinsic. What is the system, the environment that they are actually present in, whether it's species, sex, genetic background, or in context of a tumor, what kind of cancer cell pathway activation is involved, and ontogeny, where is the monocyte coming from versus the tissue-resident macrophages. And adding to this layer, you have a biological scale context that also is going to influence the fate of these cells and their ability to influence tumorigenesis, whether it's the systemic milieu that they are exposed to, the organ they reside in, the local niche, and we know those are generating a plethora of macrophage subsets that we yet don't fully understand in the context of cancer, and intracellular interactions that have. And talking about the systemic kind of biological scale, I think this is now very much central in the context of metabolic disorders, where we know that those inflammatory signals that will reach, of course, the systemic circulation, but also the bone marrow, will change the capacity of macrophages to potentially affect specific organs like the liver or the pancreas and seed the soil really for development of different type of metabolic disorders such as MASH in the liver or pancreatitis, T2 type 2 diabetes in the pancreas. And so there is this imbalance at the systemic and local metabolic level that primes the development of cancer in those organs, and that does limit cancer therapy. So again, this very large impact of macrophages and their precursors in influencing organs and later on the potential of tumorigenesis within these organs. So in the lab, we historically have been trying to overcome this one-size-fits-all challenge in targeting macrophages, particularly in two organ sites that are primarily primary tumor sites. And those cancers, glioblastoma and hepatocellular carcinoma, HCC, rarely metastasize. And that's kind of the umbrella they have of things they have in common. They are rarely metastasizing, primarily killing patients as a primary tumor. Of course, as all cancers, they are highly heterogeneous in terms of their genetic drivers. And they have this large pool of tissue-resident macrophages, the Kupffer cells in the liver, the microglia in the brain, that we originally wanted to understand how in two very different organs those tissue-resident macrophages influence different type of tumorigenic properties of these two tumor types. And of course, glioblastoma, as we know, does not respond to immunotherapy at all, to T-cell-centric immunotherapy pretty much at all. And in hepatocellular carcinoma, this response is quite limited to a subset of patients. And the type of therapies that are applied are actually much more stromal-centric than T-cell-centric. I'll get back to it in a minute about it. So what we've done in the lab is use, of course, previously generated genetically engineered mouse model, but also develop somatic immunocompetent murine models or adapt previous models that were out there in the field to our specific questions. And in order to, as best as we could, mimic the human subclasses of glioblastoma, but also of HCC. So it's not just one tumor type we could model, but really the diversity within those two cancer types. And so I'll organize a little bit the presentation around some of the main questions we try to unravel in those cancers. One being the determinant of the genetics of cancer cell, of the pathway, oncogenic pathway overactivation present in glioblastoma or in HCC particularly, that would shape the tumor microenvironment to its advantage, and how we can therapeutically exploit this influence of cancer cell-mediated genetics on immune cells. And then I'll move on talking about some of the work we've done on immune cell dynamics post-treatment, understanding what underlies these dynamics and how it actually is different based on therapy, but what we can learn from it in terms of therapeutic targeting. This will all, of course, include a lot of discussions on macrophage heterogeneity in these contexts, how this heterogeneity comes to be, what is the evolutionary need from the tumor to actually generate such a heterogeneous population of macrophage, what is this influence, and reciprocally, how different macrophage subset can influence cancer resistance to therapy or response. And it kind of led us serendipitously, I have to say, in looking at metabolic coevolution in the TME. Again, how this cooperation, competition of nutrients could actually orchestrate the interaction between macrophages or other immune cell types and cancer cells to the benefit of the tumor. And this is a lot of ongoing work we are continuing to explore. So I'll try to touch base on those different notions in the next 45 minutes or so, starting by talking about the genetic models or somatic genetically distinct model we generated in the liver to give rise to different type of murine HCC mimicking the human pathology. And so we do that using hydrodynamic tailvine injections, where we can enforce oncogenic drivers that are relevant to activate pathways that are seen in human HCCs, such as overactivation of the MAPK pathway, driven by point mutations in RAS12D or RAS12V, activation of the PI3K pathway by PTEN knockout, and a knockout of the pathway p53 pathway, which is very prominent in HCC, and overactivation of the MYC pathway. And so when we generated those different models, we first looked at whether the tumors we had that were present in the mice would have different type of penetrance or aggressivity. And we can clearly see that MYC is a quite dominant oncogenic driver that kills the mice independently, or at least to the same extent in p53 knockouts or p10 knockouts. But what was more surprising to us is the differences in just one point mutation in the NRAS oncogene, D or V, that would give rise to a relatively rapid but intermediate phenotype in survival of the animals versus the G12V tumors that are actually more indolent, longer to outgrow, and also longer to lead to the mice death. And if you look at the histopathology of these tumors, we also see clear striking differences. The MYC-driven tumors are very dedifferentiated, proliferating extremely rapidly. The NRAS G12D is highly fibrotic, which is a feature of HCC, and the G12V PTEN knockout has this accumulation of steatosis that we had not seen so prominently in other models. And that was kind of reminiscent already that it was a steatotic liver that was homing those tumors, potentially resembling the MASH HCC or metabolic associated steatohepatitis HCC human subclass. So when we did bulk seq on those tumors, what it confirmed is that indeed the MYC is a potent driver. Those tumors actually look alike in terms of their transcriptional program, whereas the 2-point mutation in RAS led to quite a segregated transcriptional signature, with the RAS12V resembling more of a control liver, which is, of course, a liver in which we injected empty vectors, so HDTV injections without oncogenic drivers. And when we took those signatures and applied them to the TCGA and other classification of human HCC, clearly now those classifications are well established. You have a lower survival, more proliferative class in which the MYC-driven tumors and this NRAS G12D tumors belong to, more aggressive HCC in patients. And then the RAS12V actually segregated into the non-proliferative, more indolent, less aggressive class of HCC. And this is actually where the MASH HCC patients sit. So when we looked at the tumor microenvironment, was really our question: how is this difference in oncogenic drivers activation impacting the TME shaping of those tumors? What we see is that the more aggressive tumors, as expected, somehow had more myeloid cell content, whereas the G12V, more indolent MASH-like HCC model, was more abundant in lymphocytes. So I'll summarize quite briefly. What we see is this G12V has more lymphocytes and mainly T cells, CD8s and CD4s, whereas the MYC-driven tumors presented very scarce T cells. There's still some in that tumor microenvironment, but that we mainly have myeloid cells, particularly neutrophils and monocytic cells. And the G12D model is the one that had this high abundance of macrophages and PD-L1 expressing myeloid cells and pretty much absolutely no T cells whatsoever. So we zoomed in into that RAS12D model because of its fibrotic histopathological features and the fact that it was so different from its counterpart that was more steatotic. And what we found is that there's this mixed background, mixed phenotype of myeloid cells by single-cell RNA sequencing that presented features both of inflammation, IL-1 pathway was overactive, for example, but also immunosuppression, TGF-beta or VEGF, were highly upregulated in these monocytic cells, particularly with regards to VEGF. And so when we looked at that, we thought maybe coming back to the cancer cell genetics, what would be secreted factors that would influence this different type of differentiation profile of these Ly6E low myeloid cells. And so we took our different data sets generated from those tumors, whether it's the cell lines we generated, the bulk sequencing, cytokine arrays, etc. And actually what crossed over is 9 either proteins or genes upregulated, particularly in NRAS G12D cells or tumors, and one of them was GM-CSF, which is somehow known as a master regulator of myeloid cell differentiation. And indeed, if you look at the cancer cells themselves, only G12D-PTEN knockout cancer cells secrete high levels of GM-CSF. And so the thing you do, obviously, when you have such a potential dependency on one factor is to try to hit it and see if you're going to impact tumorigenesis. So we did that using anti-GM-CSF, neutralizing antibody both in the HGTV somatic model and in orthotopic model of cell lines we implanted in these mice liver. And what we see is that we can somehow limit the expansion of Ly6E low myeloid cells after GM-CSF treatment within those tumors that was associated with relative tumor growth that was decreased and an overall benefit in survival as a monotherapy, which is something somehow difficult to achieve in relatively aggressive models like these. We also compared GM-CSF efficacy to the standard of care in HCC right now, which is an anti-VEGF and anti-PD-L1. And we see that if you take those therapies, they perform comparably. But if you would actually combine GM-CSF with the stronger or the more potent arm of the standard of care, which is VEGF neutralization, we can have a synergistic effect in extended animal survival. And so that tells us that actually in this subset of HCC, subtype of HCC, that represents about 15% of HCC human patients, GM-CSF could be an interesting therapeutic target by limiting this mixed bag of myeloid cells that are Ly6E low, expressing both inflammatory and immunosuppressive features. Now, if you do the same type of treatment with GM-CSF in the NRAS G12V counterpart that is highly steatotic, you don't see a beneficial effect. You actually don't see a beneficial effect either of the standard of care. And that really pointed out to the idea that here this is 1-point mutation, but in general, subtypes of HCC should probably be targeted with immunomodulation that are not the same, and that should be tailored to the type of immune environment, to the type of histopathological features present within those tumors. And so we explored a little more that HCC model we generated with G12V and PTEN knockout in terms of trying to find what could be an interesting myeloid-centric target to hit within those tumors. So, like I mentioned, this G12V PTEN knockout model does not respond to the standard of care therapy. And I just want to notice that it's not a unique feature of the G12D PTEN knockout tumors to actually respond to standard of care. This is also the case in our MYC-driven tumors, which if you would look at their transcriptional program, they resemble a bit more the viral-induced HCCs that are found in patients. But this MASH-like HCC model, let's say, does not respond to the standard of care. So we zoomed a little deeper into this model, which takes about 3.5 months to grow, and sacrificed the mice after different time points post hydrodynamic TALVA injection, so post the initial providing the initial oncogenic signal. And what we see is that after about a month, steatosis is initiated with an accumulation of lymphoid aggregate, again, reminiscent of patients. And what we see is that it's progressively increased, and the steatotic features are exponentially set up in this model, finalizing into hepatocyte ballooning, very characteristic of MASH HCC. We also see that systemically there's an expansion of triglyceride in the blood of these animals and within the tumors, and that this MASH phenotype, this transcriptional signature of this model, resembles what has been reported for MASH livers in animals. And so what Claudia and Daniel wanted to do is to explore again this idea that we could find a vulnerability in the myeloid cell landscape of these tumors that could potentially be interesting to therapeutically exploit. And so we took all of our different stages, whether a healthy liver, pre-tumor, before any neoplastic lesions are visible, or end stage when obviously the tumor burden has taken over those mice liver. And we did single cell RNA sequencing on it. And what they see, of course, is, a diversity of immune cells. And what was quite interesting is to see that the T cell pool is very much expanded, as would be expected, but so is the endothelial cell pool, and the monocytic specific monocytic subset appear to be specific of each stages, either pre-tumor or end stage. And so we looked at that in this kind of gross categories of what would be the MASH, so the metabolic disease in the liver before tumor, and in the MASH HCC. And indeed, you have an accumulation of CD8 positive T cells that are highly exhausted through time. And this was, again, interesting because we know that MASH is one of the etiology of HCC that limits immunotherapy efficacy in human patients that will have developed tumors. So zooming in into our monocytic cells, we did some pseudotime and some pseudotime analysis to try to understand what are those different monocytic cells emerging through time in this model that could have those lipid-associated phenotype. And so through those analysis, what we find is that there's an expansion of two particular subsets, CXCL16 ApoE or CD38 ApoE macrophages, that seems to be dominant in the end-stage tumors, and those are actually the ones that present the largely the most abundant lipid-associated macrophage signature that was reported by Ido Amit a few years ago. And so when you look at the lipid-associated macrophages in the literature, now you would hear a lot about TREM2. And in those terminal monocytic states in MASH HCC, in our MASH HCC model, we do see that those, particularly the IL-1 beta and the CXCL16 subsets of macrophages, are the one expressing the most TREM2. And that tells us that within this MASH HCC model specifically, we should and we are targeting TREM2 and potentially some of the precursors of those TREM2 subsets like IL-1 beta macrophages. So this is experiments that are ongoing. I can't say too much about it, but it's looking good. And so that kind of closes what I wanted to say regarding this, again, personalized type of myeloid therapy approaches in different subsets of HCC. And I'll move on and discuss a little more what we have done more in the brain ecosystem related to immune cell dynamics in general, and then zooming in into macrophages. So we all know that human glioblastoma is a deadly disease that has pretty much not changed in terms of standard of care therapy compared to the past 20 years now, where radiation and temozolomide are applied following surgical resection to the maximal extent of what the neurosurgeon can do. And yet survival is poor. T-cell-centric immunotherapy has failed. And so the idea that we had here is to look at the tumor microenvironment changes post standard of care therapy, particularly radiation, which is really what gives the most survival benefit aside from surgery, and try to see what is the phenotype of T cells that could potentially change our idea of how to target T cells in brain tumors, although they're very scarce, with the idea that when may be the new how, rather than completely changing the type of T cell checkpoint blockade to apply. And that is, a few years ago, what we have done is to look at matched primary and recurrent tumors in human patients and mapping the immune landscape using imaging mass cytometry, something we did with Daniela Quell in McGill in Canada. And what was a bit surprising is to see that CD8 T cells, and now it's been, not as surprising as it was when we got those results 5 years ago, but CD8 T cells can accumulate in recurrent glioblastoma. But they are mainly exhausted. And they come hand in hand. This increase comes hand in hand with a discrete but significant increase in regulatory T cells, so immunosuppressive adaptation in the brain tumor microenvironment. And of course, different subtypes of monocytic or microglia cells that also show this immunosuppressive program. So again, here, quantity is not quality. There's an overall change in the immune environment, but this is not enough to make T-cell-centric immunotherapy work. So already a few years ago now, it's a bit scary to think how many years ago, but when I was in Joanna Joyce's lab for my postdoc, we had used many mouse models of glioblastoma to look at those questions of macrophage changes post standard of care therapy, particularly post radiation therapy, that here is applied as a 5x2 gray fractionated regimen, trying to mimic as best we could what patients are exposed to. And what we see is that in those tumors with different genetic backgrounds, we have either a stasis or a regression even of the tumor after this fractionated radiation, but that all mice will recur within about a month and will kill the animal. What we have seen is that if we look more closely at the timing post-radiation, we actually have a slight but quite pronounced increase of T cells, general T cells. This is CD3 after radiation has been applied. So starting at day 6, the last day of radiation, that is stable for a few days and then comes back down at recurrence. So when we looked at we see those T cell influx that happen really alongside radiation treatment and applied what has been done in the clinic, which is to give PD-1 therapy at the same time as radiation is given to the patient. We also see that there's no survival benefit and actually doesn't change much compared to radiation. But now if we wait that these cells have accumulated a bit longer and apply the treatment at day 12, which we call adjuvant treatment, once there's the peak of T cell increase, we actually see that there is a slight but significant increase in overall survival. Again, suggesting that learning from what happens in the tumor microenvironment post a standard of care therapy like radiation here will teach us when we should actually apply even therapies we thought are not relevant for the disease. We pushed a little bit here and looked at the phenotype of those CD4 and CD8 T cells in the different treatment group. And I won't go into too much details. It was published already 2 years ago. But what we see is that, of course, there are some changes in the myeloid compartment, but the main change in the lymphocyte compartment was regarding the regulatory T cells. And by applying the concurrent immunotherapy with PD-1, so together with radiation, giving anti-PD-1 leads to a more rapid increase in the regulatory T cell content that are more proliferative than what we see in the group of treatment that has a slight increase in overall survival. And so here again, Tregs seemingly are important for limiting the efficacy of T cell-centric PD-1. So we blocked CD25, we use CD25 to block regulatory T cells, to inhibit regulatory T cells. It's a drug that was developed by Sergio Quesada. And here we actually can see that we can push those treated animals with radiation and PD-1 to survive better, even in the concurrent setting. And so I think what this study told us is that there's a dynamic evolution of the glioblastoma TME post-therapeutic intervention. There's a lot more that has been done in the past few years, also by many groups, showing how this is actually relevant to understand the phenotype of these cells. And so following this analysis and paralleling this analysis of the response post-therapy, we also looked at how immune cell heterogeneity would be relating to the differences seen in the cancer cell heterogeneity or the tumor subtype, the glioblastoma subtype. And so there are 4 glioblastoma subtypes, OPCs, ACs, NPC, and mesenchymal, that are usually found in the same tumor, as I'll mention later. And so what Dan and Joe in the lab wanted to do is to also look at the immune cell heterogeneity related to those different tumor subtypes. And that's what I'm going to talk about for the next 15 minutes or so, is how this glioblastoma transcriptional subtype actually shapes an environment that would be beneficial for the tumor cells to thrive. And so we started doing that knowing that human glioblastoma were transcriptionally diverse. It wasn't completely clear how diverse our own mouse models were. I need to precise, those are not our mouse models. They come from the lab of Professor Holland. who shared them with us, and then Joanna shared them with me. And looking at the tumor cells within those mouse glioblastoma tumors, either in primary setting or in recurrent setting post-radiation therapy, what we see is that we also have this variety of tumor cell subtype within our urine tumor. So it's a good model. We can use it to ask the questions we wanted to ask. One thing that is important to also know is that most of the primary tumors that can emerge with a dominance of transcriptional subtypes being APC, AC, NPC, or MES will somehow converge to become mesenchymal at relapse. And this is work from Robert Verac really elegantly show in human samples that these different classifications tend to move towards the mesenchymal subtype at recurrence. And so if you look at our mouse models in primary setting or in recurrent setting, the tumors don't become dominantly mesenchymal, but they are enriched in mesenchymal cancer cell types in the recurrent setting compared to the primary. So again, showing that we can mimic some of the features of human glioblastoma patients responding to therapy. And so why is it important? Well, if we look at, of course, OPC or mesenchymal dominant or enriched tumors, we do see that they have different features at the tumor cell level. And one of them is related to their metabolic capacities or metabolic needs. The OPC dominant tumors are pretty much, let's say, equally capable to perform different type of metabolic processes from glycolysis to fatty acid metabolism or steroid biosynthesis. The mesenchymal-enriched tumors are very much skewed toward glycolysis or TCA cycle and not really prone to generate their own lipid or to deal with their own lipid. And so that kind of called our attention because obviously this is probably a feature. They still need fat, they still need lipid to actually proliferate. And so that called our attention to look at that angle in the rest of the environment, and particularly in macrophages. And this is what we did, single cell sequencing, different primary or recurrent tumors, mouse tumors, and looking at the landscape of macrophage diversity. So as expected, we see that just the ontogeny of the microglia or the monocyte-derived cells can segregate these macrophages quite elegantly. I always find it quite beautiful to see how these cell types, which for so long we consider to be macrophages, are actually very different, and it's visible here. But what was also quite nice to see is that there's somehow a trajectory of differentiation, both in the microglia or in the monocyte compartment, where a more physiological or at least homeostatic-like cell type, like the P2YR12 microglia, will move towards becoming more inflammatory, more proliferative eventually in its programming. And same thing for the monocyte-derived cells. CCR2 monocytic cells are the ones that we anticipate are just very recently recruited to the tumor microenvironment. And then you have all these other subsets that would be natural evolution of their transcriptional program. But what really called our attention is that subset, the MG3, MDM3, that are characterized by high expression of GPNMB. And that was because in the monocytic cells, as these cells come to be, they lose their inflammatory features and they acquire a lot of metabolic features related to lipid metabolism. So we don't see that inflammatory phenotype in the microglia because the microglia are slightly less inflammatory. They're more stiff in this sense, but they also do acquire the same features of foam cell differentiation, lipid transport, lipid storage, etc. And that was something that we thought was quite interesting to see that both subtypes or subsets of macrophages in brain tumors would acquire this shared phenotype. And so this is, of course, transcriptional. We see all this lipid metabolism, but we decided to look at actually the tissue and human tissues as well to see if we could visualize these cells. And so we used BODP, which is a neutral lipid stain marker, and PLIN2, which stains the membrane of lipid droplets, to look at lipid-laden macrophages. We coined them that based on lipid-associated macrophages found in other tumors. We can see them present in the environment. We can also see many cells that are not lipid-laden macrophages. And so we first wanted to validate the fact that those GPNMB high macrophages, monocytes and monocyte-derived and microglia, had this real increased lipid metabolism activity. So we took a parallel approach of sorting cells that accumulated lipids in our different tumors, and then sorting them to perform RNA-seq, ATAC-seq, and spectral flow. And that kind of confirmed quite clearly that GPNMB high macrophages that accumulate lipid are lipid-laden indeed. The transcription education was completely overlapped. We also see using this transcriptional signature that merges both microglia and monocyte-derived macrophage, and I need to mention, could predict a worse glioblastoma patient survival and could predict a poor response to immunotherapy. So this is a patient data set of melanoma patients treated with PD-1 CTLA-4 because, as you know, there's no responders in glioblastoma, very little responders to immunotherapy. But indeed here, the presence of LLM classified non-responders. We also see and looked for whether there was a correlation with a particular subtype of glioblastoma, both in our murine samples, but also in patients, seeing the different metabolic capacity of mesenchymal tumor cells. And we found that in both cases, in mice and patients, the accumulation or the abundance of lipid-laden macrophages that have this LLM signature actually correlated with more mesenchymal transcriptional signatures in those patients. And so that led, it was really interesting to find this somehow new phenotype in macrophages. We next asked, what drives this lipid-laden phenotype? And so foam cells in multiple sclerosis have been reported, and they have been reported to be myelin-laden. Myelin being the most abundant lipid present in brain, but it's a lipid that is, of course, encapsulated in the myelin sheath and not readily accessible to cells out in the brain. That's why we always, it is always said that the brain is a very metabolically autonomous environment because all this lipid doesn't come to access with easy access to cells. So we did some electron microscopy analysis, and indeed what we see in our tumors particularly is what resembles very closely myelin sheath organization within macrophages that express here EBA1, but also within phagosomes in those macrophages in the cytosol. And what was quite interesting is that lipid droplets were found just next to this myelin accumulation, suggesting that there is a direct link between the myelin uptake in these cells and potentially the production of lipids. So when we look at glioblastoma-derived myelin composition, it's dominantly cholesterol, which is also the case for the normal non-tumor myelin. And when we did lipidomics analysis within LLMs, lipid-laden macrophages sorted from these tumors, we also see accumulation of cholesterol species within these cells, cholesterol itself, but also desmosterol. And desmosterol is an interesting cholesterol species because it actually is a very strong anti-inflammatory molecule. So we tried to trace how does this lipid-laden macrophage population come to be? What are the key signaling pathway involved in their formation? So we set up an ex vivo tracing experiment where we basically take the glioblastoma tissue, the tumor itself, as our petri dish, dissociating it and exposing ex vivo generated macrophages that are here tomato positive to the full tumor. And in that case, what we see is that the full tumor can induce this accumulation of lipid in ex vivo macrophage and also the formation of lipid droplets. When we remove the myelin from this mix from this tumor mix, using beads, for example, we lose largely the effect in forming lipid-laden macrophages. But now if we just give myelin back, the LLMs can form again from ex vivo macrophages. So clearly there's an engulfment or an uptake of myelin by those macrophages that allows them to become lipid-laden. And so we thought, what is the functional significance of this engulfment in glioblastoma? And what are the pathways that are involved that could be protumorigenic and leading to protumorigenic feature in these cells? So in the same experiment, well, in another experiment, but same setup experiment, we sorted the different macrophages that were exposed to those different features, the whole tumor, the tumor without myelin, or without the tumor, or the myelin without the tumor. So we could actually pinpoint what the myelin itself does, what the tumor, mainly tumor cells, we anticipate, but the tumor does to promote LLM formation, and how is this synergistic? And again, I'll go a bit quick. I mean, we published this paper 6 months ago, so more mechanistic-based assays are done in there. But what we see is that myelin debris in macrophages are actually mediated by the uptake through CD36 lipid receptor, and that starts dampening tumor-induced inflammation. As soon as the macrophage pick up myelin, inflammation shuts down. It also leads to a shutdown of cholesterol biosynthesis within these cells, probably because they feel this overload of cholesterol, and that shuts down the production of cholesterol itself. This is mediated through downregulation of DHCR24, which blocks the production of cholesterol from desmosterol. And that's also why we believe we see so much accumulation of desmosterol in lipid-laden macrophages. What we also see is that desmosterol activates the LXR pathway. This is something that is myelin debris specific, particularly LXR being a master regulator of inflammation as well in myeloid cells. What LXR activation also does is to push the upregulation of lipid efflux genes like APCA1 and IBCG1. And that's something that potentially could efflux those cholesterol species or cholesterol ester species coming from the lipid-laden macrophages. And the idea there is that if indeed we see lipid efflux, we should see potentially pickup of this lipid-derived species by other cells in the environment, potentially by mesenchymal glioblastoma cancer cell, which could help them grow. So this is what we tested using clickable cholesterol chemistry and exposing macrophages that were stimulated by cancer cell tumor conditioned media to uptake this clickable cholesterol on their own, wash them up, and then put them in culture with tumor cells that are either mesenchymal-like or OPC-like. All of these are, of course, derived from our mouse models. And then we could assess the macrophage-derived cholesterol uptake within the tumor cell specifically. And what we see is macrophages do not transfer the cholesterol they picked up to the OPC-like tumor cells. They only do so in the mesenchymal tumor cell. And so we thought, okay, well, that's a great, first proof, but what are the pathways that are changed? Are we sure that there is an actual processing of those lipids once they have reached the tumor cell? And so we did a series of transcriptional and lipidomics analysis in monoculture or co-culture of mesenchymal tumor cells particularly. So we took our mesenchymal tumor cells and we exposed them to myelin, also to know whether that would lead to an accumulation of lipids within the tumor cell. And we did the same in presence of myelin, but also in presence of macrophages in co-culture. So what we see in monoculture of tumor cells exposed to myelin is that nothing is pretty much changed in their lipid contents, whether it's different cholesterols, sphingomyelin, phosphatidylserine, and others. But if the tumor cells are exposed to the macrophages and then in presence of myelin, we see all those lipid species we identified in the macrophages being transferred to the tumor cells, confirming the lipid transfer experiment. And that is mainly triglycerol, sphingomyelin, and cholesterol, again, potentially fueling the need of lipids of these cells. So is this the case? Is this going to promote different type of protumorigenic features in tumor cells? This is what we tested next. If we take mesenchymal tumor cells and put them in culture with macrophages in presence of myelin, we can see an increase in overall proliferation. And if we limit the uptake of myelin by the macrophages using a CD36 inhibitor, or we limit the efflux of the lipids from macrophages to the tumor cells using an APCA1 inhibitor, we can compromise this pro-proliferative effect LLMs have on mesenchymal tumor cells. And this is reflected in preclinical setting when we combined radiation therapy with a CD36 inhibitor. Couldn't use the APCA1 inhibitor, it doesn't cross the blood-brain barrier. We could see a modest but significant and quite reproducible effect in increasing overall survival, suggesting here again that LLMs could be good therapeutic targets if we harness them, particularly when they are very abundant, which is in tumors that are recurring, that are relapsing, that are more mesenchymal-like. So to wrap up that part of the story, what we see is that there's a large amount of myelin debris that actually do not support tumor cell proliferation by itself. What needs to happen is that the macrophages pick up this myelin debris, become lipid-laden, and that actually pushes their ability to become more immunosuppressive, which is something I haven't talked about, but also to store these lipids and activate the LXR pathway, dampening their inflammatory features. And then macrophages can mediate lipid efflux, and this fuels mesenchymal-like cancer cell proliferation, potentially participating to regrowth of or to an advantage, selective advantage for mesenchymal cells to grow and potentially repopulate a relapsed tumor. So this is what we found. I want to point out that, of course, we were extremely happy about this story, but we have been extremely inspired by developmental biology and really brain neuroimmunology, where macrophages, microglia particularly, have been known to participate to demyelination and remyelination and are main actors of maladaptive myelination, for example. So this activation profile, the change in the inflammatory features of these cells have been known in the MS and the Alzheimer's field. And here again, this is an example of the intrinsic organ physiology capacity of cells like macrophages to be hacked into by tumor cells for their own advantage, both for proliferation, but also for immunosuppression. So where do we go from there? Well, our CD36 inhibitor trial and in vitro, ex vivo assays really showed us that there is potential in targeting lipid-laden macrophages, but we clearly see that the therapeutic effect is not to our, liking. We would like to push that more by being more specific when we target lipid-laden macrophages. So we've set up collaboration with drug delivery expert and chemist to generate lipid nanoparticles and liposomal formulation that would take advantage of the fact that those lipid-laden macrophages love to eat lipids, and lipid nanoparticles are lipids. And we now have a few formulation, one particularly that looks really good in a specific uptake by lipid-laden macrophages and but not by the other macrophage subsets. And so we're testing different drugs encapsulated within those lipid-laden macrophages to see if we can actually push the effect that we had seen therapeutically. And just to show you, we've done a lot of tests of different formulation, and we've done that by generating glioblastoma in fish. That was a new area in the lab. It's still a new area of research in the lab. And this is just a very cool movie, I thought, where those PAP3 macrophages actually love to bind to myelin within the brain environment. And this is through this binding that they get encapsulated by macrophages that then become lipid-laden. You see it here very well. So it's somehow an idea of like weaponizing myelin, weaponizing the fact that those macrophages love to pick up myelin to be able to give them some therapeutics. Another area of research is a bit more fundamental, trying to understand how LLM heterogeneity, how large lipid-laden macrophage heterogeneity is, and how different niches may be leading to different phenotypes of lipid-laden macrophages. We need to understand if we want to hit the right one. So we're doing a lot of spatial proteomics and transcriptomics, so spatialomics approaches, looking at particularly hypoxic niches. Because this is in hypoxic niches, we see the lipid-laden signatures to be the more predominant in Visium sequencing. And this is also where we see indeed LLM accumulating, but also where we see their interaction being the most significant with mesenchymal-like tumor cells. And so Menno in the lab, a PhD student, has started to develop this project a year or two ago, by injecting, for example, pimonidazoles, which is a hypoxia tracer to our brain tumor-bearing animals, and then looking at macrophage populations in PIMO high or PIMO low areas. And he's doing that also by immunofluorescence, but I just wanted to show you something that was somehow surprising. I mean, we knew from the transcriptomic analysis that likely LLMs would reside in hypoxic niches. And here I'm talking about all LLMs, microglia and monocyte-derived macrophages together. That's indeed the case. But then if we look at the ontogeny of lipid-laden macrophages in normoxia, they are mostly monocyte-derived. So the monocyte-derived macrophages both and then the microglia lipid-laden macrophages are present in normoxia. But in hypoxia, it's largely the microglia that are LLMs that reside within these areas, suggesting that those microglia that are LLMs may be the one interacting the most with mesenchymal tumor cells in those very harsh environments of potentially around necrotic areas, which are feeling brain tumors like crazy. And so we're trying to model all of this and look whether there is a gradient of changes of potential accumulation of lipids through the brain. We've done that, but we're doing that using brain slice assays where we can cut pieces of our tumors, and I hope eventually human tumors as well, and add ex vivo macrophages that we can then linear trace within this live tissue growing for about a week and maintaining the spatial organization. And so here we're talking about bone marrow-derived macrophages that are TD tomato positive, and it kind of confirms what the in vivo data show, where the lipid-laden macrophages that are monocyte-derived, like to become and to pick up lipids, to pick up myelin likely around vessels. And so we plan to do the same with microglia, primary microglia derived, and understand how this all organizes within the brain environment. And so another follow-up study, again, quite fundamental, is we hopefully will have the right toolboxes to hit lipid-laden macrophages, and we hope more than lipid-laden macrophages later on. What are we going to encapsulate within those liposomes or those lipid nanoparticles? So what Martina in the lab is doing is actually a pretty ambitious CRISPR-Cas9 screen on primary monocytes and microglia from animals that she's customizing a CRISPR library customized for metabolome and surfaceome, but that will be particularly focused on understanding how we can change the inflammatory phenotype, so the immunosuppressive phenotype of LLMs, pushing it back to being more inflammatory and maybe a bit more immune activating. So she's doing that by multiple. She just spent a year literally optimizing the screen, and it's now hopefully going to run very soon. And we're quite excited to be able to present new or to identify new targets for those macrophages that have picked up myelin and apply them using the new toolboxes we're setting up with our colleagues chemists. So that's what I wanted to say today, and I just want to have a maybe overview of the different notions that I try to advance. One is that, I mean, that's not mine. Cancer is a wound that never heals. We know that, and there is this flow of recruitment of immune cells, but also mobilization of local immune cells that is going to affect, of course, education based on when they come into the organ, the state of the organ, the state of the probably the individual itself. And so what we've seen is that there's clearly an influence of oncogenic signaling that instruct immune cell and myeloid cell features. So we need to consider genetic diversity and tumor subtype as one element that fuels diversity of myeloid cells. We also see that there's a treatment-induced selection, and that can actually reveal some exploitable vulnerabilities to apply in the immune modulation field. And again, that will likely be specific to each tumor type or subtype. And of course, this view of macrophages harnessing their inherent capacity in the brain to pick up myelin is showing us that there is a deep co-option of organ physiology in macrophages, and that usually here in the case that we identified, it's to fuel cancer cell energetic needs, but it could be much larger than that, whether it's for invasion or other aspects. And there's adaptation to cellular niches, particularly in hypoxia. We believe the metabolic rewiring is at its apex in those niches. And so we need to understand a bit more about this niche organization of myeloid cells to be able to specify their targeting in different cancer ecosystems. I'll stop there. I want to say thank you to, of course, all the people that have participated to this work, the alumni in the lab. The LLM story was really led by Dan and Joe in my lab. I tried to mention everyone, the liver team, Serena, Christelle, Daniel, but all the entire group who really are a fantastic bunch that I'm very privileged to work with. All our collaborators on the liver and the brain side. And of course, all our founders, and I'll be happy to take any questions.
Thank you, Leila. It was great presentation, very educational, and I hope it will be very interesting for people. Please, if you have questions, put it into the chat. I see one question which I will absolutely certain to make sure I will ask, but I have related question. I have several, but I will start with one, which is related to the following question from Filippo. My question is the field of has been haunted by this idea of targeting individual scavenger receptor. And the problem with all these approaches is high redundancy in scavenger receptor activity. That's why it's not therapeutically feasible. My question to you is, in your situation, in this particular model, do you have evidence that CD36 is indeed the dominant, prevalent scavenger receptor which mediate myelin uptake and but other activities?
No, what we do see is that there's an upregulation of CD36 expression after myelin's been picked up, so probably pushing more uptake of myelin. We see hitting it, of course, has some effect, but we also see that TREM2 actually in those macrophages is very high, and that TREM2 also is one of the myelin
Yes, and TREM2
receptors.
has issues too in the
Indeed.
therapeutics.
So
Yes,
yeah, no,
so
I
that's
think this is a good point. Of course, you do with the tools you have, and CD36 is what we had, but I think that there will be immense redundancy. I don't think that
okay.
just blocking the receptor will be giving us sustained effect. We see some effect, but it will rapidly be overcome and compensated for.
Okay, well, that brings me to the question in the chat from Filippo Veglia. It's he said, I was wondering if the absence of CD36 on either MDM or MG may revert the phenotype macroglia and macrophages. Just, I guess I spell out the abbreviation. May revert the phenotype observed. Moreover,
Yeah,
did you take a look at other myeloid cells like dendritic cells, neutrophils with the function?
Yeah. So I do believe that if you hit only one or the other one compensate. And so the CD36 in monocyte derived or in microglia, the other cell type can probably pick up the slack. Because what we did is we did some adoptive transfer, not using flux CD36 flux mice, but lysem-cree APCA1 ABCG1 knockout mouse that were provided to us by Brian Ruffle, actually. And what we see if we do adoptive transfer, so all the macrophages that are in those tumors will not be able to efflux their lipids, we actually don't see significant increase in overall survival, because then we see that the microglia LLMs, and you have more microglia LLMs, and the microglia LLMs start to upregulate quite dramatically APCA1 and ABCG1. So they'll probably efflux even more and pick up myelin even more.
That's
So
wonderful.
this is probably, again, the issue with specialized function are important, but it's also something to consider because the other subsets of macrophages can take over. Now, for CD36, at least in our hands, in our tumors, in the brain tumors model we don't see CD36 expression in the neutrophils. We see it in the endothelial cells. So we had to do a lot of controls, trying to make sure that the CD36 targeting strategy we employed did not change anything about the endothelial cells. They express relatively low levels, so that's okay. But I cannot completely exclude it. And the tumor cells did not express it.
Okay. Zelani, you have a question, right?
Yes. Hi, fantastic talk. I love the images, the cartoons that were meticulous. My question was regarding the metabolism of these macrophages. Have you all looked at the the TCA cycle of these macrophages after they they go from being glycolytic to oxphos? Have you followed their metabolism changes as the scavenger receptor expression increases on these cells?
It's a very good question. The quick answer is no, but I'll try to explain a bit why. So we, at least transcriptionally, and again, you're looking at ataxic, we don't see too much changes in the mitochondrial metabolism, which was a bit of a surprise to us. We have not done the test, the Akkar and the different metabolic tests. This is something we have to do, but we didn't focus too much on the macrophage metabolic state in that study. We looked mainly at what the tumor cell metabolism will be, or how is it changed after this interaction with lipid-laden cells. So thinking that the macrophage at that time, and it's probably a bit of a shortcoming on our side, we should have looked at that more, but that they would just be a reservoir for producing lipids and then spewing it out to the tumor cells for growth. So what we looked at is what happened to the tumor cells. Where are the lipids coming from macrophages going when in co-culture with tumor cells. And this is where we looked at TCA cycles. So we see that part of this lipid is actually fueling the TCA cycle, but this is relatively minor, actually. It's mainly building up membranes. So we see that the bulk of the lipid transfer to the tumor cells is essential for building up membranes. So we think this is, of course, an important step in proliferation. But, I want to we started dabbling into metabolism, about 3-4 years ago. This is really something we're exploring more and more now.
Yeah, I'm curious just about the lipases changing in the cell after they have to digest it, because once those lipids are broken, engulfed by ApoE1 phagocytosis processes, lipases can break down lipids, and those lipids can then enter the TCA cycle as acetyl-CoA. And so I would think that that's probably what's occurring. I know that happens in normal cells and even in bacteria, but I was just curious if that happens
no, it's a
here
good
in the
point.
TAMs.
So Dan, who led that story, also had that question. He used a few lipase inhibitors, but at least in vitro, it didn't really change what happened to the uptake of myelin or the production of and the efflux of lipids. But I think we're probably not mimicking everything in vitro as well. This is, of course, a limitation we should all be aware of. We try our best to have the best ex vivo system possible, but it's probably not completely enough. But it's a very good point. We are looking more into it. We're particularly looking at what happens in terms of the length of lipids, the length of the carbon chains once those lipids are formed, and see how this actually influences the transcriptional status, but also the immunosuppressive status of the cells. So hopefully I'll
May I
tell
ask you, okay. I want to follow up on a little bit more functional consequences of this event from the slightly different kind of point of view. Eating myelin, pieces of myelin by macrophages and or glia is really a fundamental function of the cells. That's how they protect us from inflammation in the brain under normal conditions. And immune suppressive microenvironment is actually a good thing for brain, because that would be a disaster if it was not the case. Actually, this is similar we have in lung. My question is, how much, let's say, optimistic view is to interfere with this process in cancer with hope to change this outcome if in macrophages in this microenvironment supposed to be immunosuppressive. That's their role. And what exactly we're trying to achieve if we target them in that fashion.
Yeah. So indeed, I think most of macrophages, myeloid cell function that we see in cancer are just hack of their inherent physiology. I mentioned that. This is why I think we have to be very careful with systemic therapies. We really have to try to find a way, a bit like with the lipid-laden cells, their ability to pick up so much myelin in cancer is something we can try to use to our advantage for delivering lipid nanoparticles. Systemic therapy can lead to side effects if we don't pay too much attention to that quite quickly. Now, in the brain, microglia are actually second in line for myelination control. They can do it, and they do it particularly during development. But in a more homeostatic state, it's really the OPC and the astrocytes that have the leading role. So I wouldn't be too worried about the cognitive effect. But if we think that, and I do think that macrophage therapy is something that we should see as a very long-term therapy for it to work sufficiently, you also don't want to push your luck too much and hitting too much immunosuppressive pathways that at the same time will disrupt tissue homeostasis. I think this is very valid in the liver as well, which is a very immune tolerant organ where zonations of macrophages have very unique functions, and they're all related to controlling, not getting too much T cell crazy. So we can't do everything we would want if we think about patients.
Thank you. So I have another question in the chat. I suspect it from Sven Brandau, but it looks like, so I apologize if it is not. From a very fundamental therapeutic view, there are two parallel mechanisms, differential recruitment or presence of cell type versus functional reprogramming in response to versus responders versus non-responders. Both mechanisms require different therapeutic targeting. What would you target with priority? What will be your priority? Or is it always necessary to target both mechanisms at the same time? Which would you make therapy quite complex?
Mm-hmm.
Loaded question, I guess.
It's a lot of questions. I'm trying to organize how I can answer it. Um, I think if I understand the question, it's more what do you reprogram and rewire versus what would you limit the recruitment of?
Yes, that's basically, if I nail down, I think the question is recruitment versus functional reprogramming.
Yeah. I mean I am a bit more towards reprogramming because recruitment again becomes a much more systemic approach, and we have to be careful with what we do systemically. And the problem is that you hit what happens in terms of recruitment, so you limit, CCR2 inhibitors. They've had some success, but the problem is that the body, the cell, the environment will make more of it. And if you limit recruitment, you can actually boost somehow proliferation locally. So I do think that the recruitment in cell, I don't know what your take on that is, Dmitry, but we see, at least in brain and liver, a lot actually happens locally. So the cells are relatively naive, relatively virgin in the circulation, particularly for brain. For liver, it's not exactly true, but for brain, they only get their phenotype once they get there. And so
it depends what myeloid cells you're referring to, right? If you're talking about macrophages, they don't really travel. They got
exactly.
whatever the people think.
Yeah.
But neutrophils are, and if you target neutrophils, there is argument to be made, very strong one, that recruitment may be very important.
Yeah.
For macrophages, I would share your view too. It's just
because they're very,
yeah, you can block monocytes. Yes, you can block monocyte, but it's only part of the macrophage community. That's only
exactly,
and sometimes
my
even smaller part.
anticipation is then that they would locally find a way to be more numerous and proliferate.
Yeah.
So do we have to do both? Ideally, or my view would be actually to do, if both approaches would be beneficial, hit recruitment at the right time, hit reprogramming at the other time. Because there's a notion of timing that I feel like sometimes myself too are overlooking. We cannot hit the same thing at the same time and expect that it's going to work. We need to map out, really have this evolution view like people have had in ecology or in cancer itself, but have it applied to the immune landscape, to the tumor microenvironment. There's an evolution that underlies properties of these cells, and this is where maybe when there's specific boost of recruitment coming from the tumor, you may want to hit it, but then the reprogramming approaches come when the tumor evolves and needs it, and this is what we need to identify to neutralize.
Absolutely. Well, there's one issue which we shouldn't forget is toxicity. Each of this targeting strategy comes with their own toxicity, and sometimes pretty significant. Therapeutic effect of CXCR2 could be quite substantial, but it's also associated with neutropenia. Macrophages, the repolarization may be associated with quite a lot of side effects. So it's
and I think this is also why we opened up a bit in the past couple of years, working with chemists who have a breadth of tools that I had no idea existed. Of course, lipid nanoparticles, but you don't know how much you can modify a lipid nanoparticle to be subset specific,
selective,
selective, but also we're moving on this ERC synergy project to try to do drug conversion with in vivo catalysis, where it would only occur in macrophages or in LLMs or in tumor cells, and then we bypass the toxicity because we only give prodrugs systemically. So the drug will only be active in macrophages once it reaches the macrophages. So all these chemistry-centric tools, I think, can really open up cancer biology treatments that we just need to be open to learn new things for sure, but also to apply things that, we didn't think could actually be applied, and reached out to cross-bridge with the chemist and the drug delivery experts. There's a lot of potential there.
That's right. Well, I keep talking because I try to give time to people to put these questions, but I will ask last question with hope maybe someone will ask another one, because right now we don't have any questions in the chat. If you, I would like to bring you back to your first part of your presentation. You provide some quite exciting data with GM-CSF targeting and the GM-CSF producing subset of macrophages. And this part looks fine, but with working with GM-CSF before, it's kind of what question I'd like to put on you. Your clinical analysis, do you have data or what data you have that GM-CSF is associated? First of all, there is a substantial proportion of patients who do produce a lot of GM-CSF generally. And the second one, how much that connected with the clinical outcome?
Yeah, so we do have clinical correlations where patients that would have, so the way we generate our model with this overexpression of NRAS point mutations, NRAS G12D will have a quite strong MAP kinase activity versus G12V having a lower MAPK output. So what we did is like we mapped the levels of ERK1 and 2 and MAPK activity in TCGA HCC patients and looked at the ones that were somehow matching what we see in the mouse in the G12D model. And if you look at this group of patients, and it's in the paper, those are the ones actually that have correlative GM-CSF signature within their tumors, suggesting that they do produce, or at least the tumors show more GM-CSF in their environment. In terms of prognosis, we didn't see anything particular. We only see that we only did the analysis based on our murine signatures, and looked whether the patients associated with that signature or vice versa actually had differences in survival. And indeed, that's what we see with the G12D patients having this more intermediate survival benefit compared to patients that have very aggressive MYC-like tumors or the G12V tumors. We didn't link it exactly to GM-CSF. That's something we could
Right,
do.
because I would assume that if the subset is at play, on the patients probably don't care about subset of macrophages. If GM-CSF is really driving force, you should see link with the transcript generally, unbiased way.
We should definitely look at that.
Anyway, it looks like we exhausted, first of all, our time. It's already 12:12 in East Coast. And then no questions. I would like to thank you very much for your presentation. Thank you, everyone who's still on the call. We have quite a few people. And looking forward to the next meeting. Thank you, Leila.
Thank you so much, Dmitry. Thank you, everyone, for the discussion. And have a great day or evening based on where you are. And see you next time.
Bye.
Bye.
Bye-bye.